Hello, my name is Joy Mendel. I'm part of the Gene Lab, and I'm presenting my research on the investigation of flagellar biosynthesis differential gene expression in a delta-long Caulobacter crescentus. So just to give a little background, Caulobacter crescentus is an aquatic gram-negative alpha proteobacteria that can be found living in bodies of freshwater and saltwater. This organism serves as a critical model for understanding cellular polarity, cell cycle regulation, and cell differentiation. An elaborate network of signaling and regulating proteins coordinate the Caulobacter asymmetrical cellular division to yield two cell types. These types differ in both their morphology and function. The first type is the modal Swimmer cell equipped with a polarized flagellum, and the second being a sessile stalk cell equipped with a polarized polysaccharide base adhesion and known as a holdfast. Stalk cells can initiate replication immediately, whereas it is pertinent for the Swimmer cell to shed their flagellum and produce a holdfast before they can undergo replication. During this investigation, C. crescentus strain NA1000CB15N will be studied. A wild-type version will be analyzed alongside a LON mutant in order to understand the various impacts knocking out the LON protease has on overall cell motility and viability. The LON protease functions as a primary post-transcriptional regulatory pathway for the degradation of misfolded or damaged proteins. LON-dependent proteolysis targets several primary cell cycle regulators for spatial and temporal degradation. The protein's presence is not necessary for the viability of C. crescentus, however, its removal results in significant phenotypic defects. Our mutant has the LON protease knocked out of the genome, and without it, C. crescentus has been observed to have an elongated cell body, loss of motility, and poor stress responses to DNA damage and protein misfolding. The loss of motility detected in the delta LON mutant could be explained by significant differential expression of the flagellar biosynthesis genes that I visualized. The Caulobacter cell cycle consists of three main phases, G1, S, and G2. The stalk cells spontaneously start in the S phase, meanwhile Swimmer cells are in G1. Morphological changes at the cellular poles are coupled with other cell cycle factors that together serve as a progressional checkpoint for the cell cycle phases. In G1, the flagellum is ejected from the membrane, the holdfast forms in its place, and the stalk body elongates. In the S phase, DNA is replicated, methylated, and the chromosomes begin to segregate. In addition, the flagellum apparatus also begins to assemble at the pole opposing the holdfast. Lastly, in G2, the flagellum biosynthesis undergoes completion, and the chromosomes finish segregating so the cell can undergo division. All three of the described stages are mediated by four conserved proteins, CTRA, DNAA, CCRM, and GCRA. It is critical for both the timing of flagellar shed in the predivisional stages and flagellar biosynthesis in the late predivisional stages to occur in order to have successful polar morphogenesis of C. crescentus. Flagellar genes are expressed as a four-class hierarchy. Class 1 proteins are produced before class 2 proteins can be produced and assembled, so on and so forth. CTRA is an essential transcription factor that has the ability to both repress and activate these polar morphogenesis genes depending on its own cell cycle regulated phosphorylation and interaction with protein CyP. CTRA is produced and activated by the temporal promoters P1 and P2 and is recruited to the flagellar pole as it actually represents the first class of flagellar genes in the hierarchy. C. crescentus is robust in its ability to have several different layers of control in case one fails. So P1 can be activated in predivisional phases by the hemimethylation of N6 adenosine so that GCRA can bind. And it is deactivated by either the full methylation of the promoter or by the binding of CTRA as a form of negative regulation. The P2 promoter undergoes positive regulation in which the activated CTRA acts as an activator to turn up its own transcription of the gene. And the promoter is repressed by an inhibitory complex consisting of CyP, CTRA, and a phosphate. All right, so CTRA has a phosphorylation site at the asparagine 51 residue. Phosphorylation at the site is achieved by the bifunctional enzyme CCKA. The addition of this phosphate sustains activation of the CTRA protein and increases its binding affinity to the target regions of the DNA. Activated CTRA can bind to CyP transcription factor to create an inhibitory complex that I mentioned earlier. The histidine kinase DFK phosphorylates PLD, allowing for subsequent ejection of the flagellum and stalk elongation. PLD concentrations dictate cyclic diguanosine monophosphate oscillations and behaves as an allosteric effector by binding to the domain CCKA and creating a conformational change that results in CCKA switching into a phosphatase. This phosphatase then removes the phosphate from CTRA, rendering it inactive. Deactivated CTRA can undergo degradation by the ClipXP protease. As I mentioned earlier, CyP is an essential layer of control over CTRA. The protein is a helix-turn-helix transcription factor that binds directly to the phosphorylated form of CTRA. It is also a substrate of the LON protease. Once bound, CyP interacts with CTRA to create an inhibitory complex. The formation of this complex serves to intervene and prevent untimely activation of certain genes throughout the cell cycle, and therefore the CyP levels oscillate alongside alongside CTRA's concentration. The principal function of the regulatory complex is to bind to target DNA and prevent the recruitment of the RNA polymerase. If the RNA polymerase cannot establish a position with this motif in the promoter, then translation of the gene cannot occur. This complex has the ability to bind to all CTRA target sites except the 5 that lie within the origin of replication. So now that we have a grasp on the background, these are the questions I am looking to answer. So my hypotheses, my 2 running hypotheses, are that the flagellar biosynthesis genes are downregulated and conserved within a delta-1 mutant due to the cell's inability to degrade CyP. And then my second hypothesis is that FLHA and FLHB are synthetic lethal in a delta-1 mutant because they are both transcribed and assembled as a pair. Other paired flagellar biosynthesis genes are predicted to display the same lethality patterns. So for this investigation, raw data from the RNA-seq, TN-seq, proteomics, metabolomics libraries were developed by the Gene Lab and were used. This data was obtained and presented for both the wild type and the delta-1 mutant, and are visualized using the IGV and Excel platforms for analysis. In addition to these libraries, statistical information on these data sets was also utilized. The RNA-seq data provided a general genomic annotation on whether or not the genes were being differentially expressed by yielding the relative transcriptional counts. Differential expression can be manifested by either the up or down regulation of the gene in the delta-1 library. Upregulation was hypothesized when expressional levels of a gene seemed to be increased in a delta-1, and downregulation was hypothesized when the expressional levels seemed to be decreased in a delta-1. The TN-seq data similarly provided a general genomic annotation, but in reference to how essential the gene was to the cell, given their relative transposon mutagenesis. Gene essentiality can be assessed by examining which genes can and cannot tolerate transposon insertions. Gene regions with high quantities of transposon inserts would indicate that the region is suspensible, and loss of function in this region would likely still yield a viable cell. A lack of transposon insertions would indicate that this gene is essential for cell viability and therefore not dispensable. It was hypothesized that delta-1 genes with few to no insertions compared to the wild type had to have become conserved, and the raw RNA-seq, TN-seq counts underwent a statistical analysis to prove this. The log FC values were analyzed for positive and negative fold changes, and each of these points also generated a p-value. The p-values below 0.05 indicated that there was strong evidence supporting the log FC value, and values higher than 0.05 indicated that there was no statistical significance. FDR values were also determined and indicated confidence in this statistical significance. Genes of interest and their relationships to other genes were further explored using the string databases, Fitness Blast, Microbes Online, and then were further interpreted using RStudio. The networks manifested by the flagellar biosynthesis genes were used as a primary model in investigating CyP and CTRA influence. So it is known that the delta-1 mutants show fitness defects such as motility loss. Therefore, in order to determine the impact of removing the LON protease on flagellar biosynthesis genes, the RNA-seq and TN-seq genomic libraries were analyzed on the IGV platform. The first thing I was able to determine in my research was that the FLHA and FLHB genes appeared to have become both downregulated and essential in a delta-1 background. Statistical measurements confirmed the downregulation and conservation to be significant, suggesting that the LON protease is indeed needed for proper flagellar assembly. The bottom 2 tracks for both FLHB and FLHA highlight the relative transcriptional frequencies for both the delta-1 in pink and wild type in blue, and the upper 4 tracks for both FLHA and FLHB display the relative transposon insertion frequencies for both delta-1 again in pink and wild type again in blue of C. crescentus. During the characterization of this result, we noticed significant patterns of downregulation for all of the remaining flagellar biosynthesis genes. Additionally, the inability to tolerate transposon insertions was witnessed for all remaining flagellar biosynthesis genes. And so this downregulation you can see on the left to this dotted dashed line. All of the blue dots you see here are the flagellar biosynthesis genes. And if they are in this red region on the left, then that is that they are both downregulated and that downregulation is significant. Genes of interest are highlighted in green. The newfound essentiality of flagellar biosynthesis genes in a D-LON can be explained by the upregulation of CyP. The LON protease handles eliminating the CyP proteins that have already been produced, and phosphorylated CTRA activates both the transcription of CyP and of the class 2 flagellar biosynthesis genes. Throughout the duration of the S phase, it is critical for the LON protease to degrade the produced CyP so that it does not form an inhibitory complex with CTRA. And with upregulation of CyP, it's suspected that there's not only an increase in this frequency of inhibitory complex formation, but also the untimely formation that is turning down the expression of genes that should ordinarily be expressed. The degradation of either protein by a protease would lead to the cooperative disassembly of the complex, and within LON, CyP cannot be proteolyzed and therefore not removed. However, CTRA is a substrate of ClipXP, which is still there and therefore can still be degraded within a delta LON mutant. However, because flagellar biosynthesis genes are still visualized as being turned down, then this disassembly is probably not occurring. The binding of CyP to CTRA stabilizes CTRA from proteolysis by ClipXP. CTRA in its active form has a high affinity for CyP, therefore the upregulation of CyP in a delta LON stipulates that in an environment with more available CyP, then there's going to be increase in the formation of this inhibitory complex. Slow disassembly rates combined with increased inhibitory complex yield circumstances in which genes typically activated by CTRA are experiencing more inhibition. So studies conducted prior revealed that amino acid residues 35 and 40 are critical in maintaining the tight bind between the two proteins. These residues form a recognizable region for ClipXP to recognize CTRA as a substrate and degrade it. When CTRA is bound to CyP, regions of the CyP protein may be either obscured by or is no longer interacting with this recognizable region in such a way that it's no longer accessible to ClipXP. It's also plausible that when CyP is bound to CTRA, CTRA undergoes a conformational change that causes the recognizable region to shift into a non-accessible position. If CTRA is inhibited at the class 2 stage, then class 3 and class 4 genes will remain unexpressed, as you see here. So given that this is only one semester's worth of work, more time sifting through the data would likely confirm upregulation of CyP in the delta LON mutant does indeed lead to the downregulation of flagellar biosynthesis genes. I'm currently still in the process of investigating the synthetic lethality of FLHA and FLHB. The deletion of either gene on its own in a delta LON background does not lead to any critical deficits in the organism's fitness. However, knockout of both of these genes create a non-viable organism. This means that the dual knockout of the FLHA and FLHB are synthetic lethal in their consequences. The two genes together help form an export apparatus for the flagella to pass through the gram-negative secrescentus thick peptidoglycan layer. And these two proteins together form large cytosolic domains for substrate interaction. These genes already have decreased expressional levels, but I'm working to determine if their lethality when removed stems from either the failure to meet a critical cell cycle checkpoint or if there's a different underlying cause. Additionally, I'm investigating other flagellar biosynthesis genes for relationships that could yield synthetic lethality when they were both knocked out. Future research would be to investigate the increased presence of CTRA due to avoiding degradation by ClipXP through binding of CyP, and if that is correlated to the upregulation of CCKA. Upregulation of CCKA should be correlated to an upregulation of DivK and PLD. So the raw sequencing data, statistical analysis, pattern confirmation in R, CyP knockouts, flagellar gene knockouts, protein concentration detection using proteomics, as well as literature review, can all be used as tools for this future research. I just want to give a special thank you to Dr. Chin for allowing me to join the lab at the beginning of my senior year, and he's given me endless support and motivation and encouragement to an otherwise hectic semester. To Mr. Lauren for never-ending patience and wisdom and advice. Aurelia, someone I work in the lab with who has been nothing but motivating throughout this entire experience. And lastly, my friends and family. If you have any questions, please feel free to email me at jmendel@umass.edu, and you can attend my question session at April 26, 2 to 3 p.m. via Zoom, RSVP for a spot. Thank you.